Welcome back. In this video, we're going to look at the degradation pathway for the catecholamines. And in this particular example up here, this I got off of Wikipedia, we're going to look at the degradation pathway for norepinephrine. Of course, we can do it with epinephrine, and we can do it for dopamine. And the good news is that if you're learning this degradation pathway, they're all going to follow in the same suit. Okay, now initially, there are two enzymes that can react with norepinephrine. One of them is referred to as catechol-O-methyltransferase. And what this enzyme is going to do is it's going to catalyze a bimolecular substitution reaction in which a methyl group is donated from S-adenosylmethionine onto the hydroxyl group that's meta to the beta carbon. So notice that if we react with catechol-O-methyltransferase, we get this methyl group on the hydroxyl group that is meta to the beta carbon, right? Or we can say it's ortho to this hydroxyl group, right? So catechol-O-methyltransferase catalyzes the SN2 reaction of the methyl group transfer from S-adenosylmethionine to norepinephrine. And we can do the same thing for epinephrine, right? It's just that in epinephrine, we have a methyl group that would be attached onto this amine, right? On this amine, we would have a methyl group in epinephrine, and if we were dealing with dopamine, the beta hydroxyl group would not be there, right? Because dopamine has not reacted yet with dopamine beta hydroxylase, okay? And if you need more review on that, certainly go watch the catecholamine biosynthesis video, okay? Now, for complete inactivation of the neurotransmitter, we have to react with another enzyme, and that's called monoamine oxidase. And monoamine oxidase is an FAD-dependent oxidase, and what it's going to do is it's going to catalyze the oxidation of the neurotransmitter into an aldehyde. So notice that in all of these degradation products, both normetanephrine aldehyde and norepinephrine aldehyde, notice that we have this aldehyde that's ultimately part of the alpha carbon, but it's basically attached to the beta carbon, right? So we have an aldehyde that's characteristic of both of those. And for complete inactivation of these neurotransmitters, there's an absolute requirement that you metabolize the neurotransmitter with monoamine oxidase. So for complete inactivation, you have to use this enzyme, okay? To further inactivate it, you can use catechol-O-methyltransferase, but that's not an absolute requirement. The absolute requirement for total inactivation is monoamine oxidase, and we'll see why that is in a few minutes. Now, one thing I want to mention about these two enzymes, catechol-O-methyltransferase and monoamine oxidase, is that in both cases, the norepinephrine or the epinephrine or the dopamine, whatever you're talking about, can react with both of these enzymes in either order. Well, what does that mean? Well, you could have norepinephrine in this case, and it could first react with monoamine oxidase and then react with catechol-O-methyltransferase, right? Or it could react with catechol-O-methyltransferase first and then react with monoamine oxidase. Okay, so if you were to react with both of them, there's no preferential order in which they have to react. Okay, but if you want to completely inactivate the neurotransmitter, there is an absolute requirement that monoamine oxidase react with it, and let's explore why that is now. Well, keep in mind that an aldehyde, if you were to look at an aldehyde, right? Let's say we take acetaldehyde, right? Here's an aldehyde. If you were to go, you can oxidize this or reduce it, right? So it can go in either direction in terms of oxidation states. If you were to reduce an aldehyde, what do you get? Well, you just get an alcohol, right? You get a primary alcohol. Whereas if you were to oxidize the aldehyde, you're going to get a carboxylate at physiological pH, right? So the aldehyde can react with various enzymes, and there's two of them here, and it can either reduce the aldehyde down to an alcohol, or it can oxidize it up to a carboxylate. Now, what I want to make perfectly clear is that the naming of these products is not really the most important thing. What's important is being able to predict what the product looks like based on the enzyme that reacts with it. Now, what we're going to assume in this case is we're going to make the assumption that we've already reacted the neurotransmitter with monoamine oxidase. And so we have this aldehyde that can be reacted either with an aldehyde dehydrogenase or an aldehyde reductase. Now, what's going to change in these reactions is whether or not the hydroxyl group that is meta to the beta carbon has been methylated. So notice that in normetanephrine aldehyde, that hydroxyl group that's meta to the beta carbon. Notice that this right here, this is the, oops, let me get my brush. Oops, wrong color. Notice that this carbon right here is the beta carbon, and notice that the hydroxyl group that is meta to that has been methylated. But notice that in norepinephrine aldehyde, notice that, oops, let me undo that. Notice that this hydroxyl group has not been methylated. The one that's meta to the beta carbon has not, it's not been methylated. So that's also going to dictate what product we get. And so if we start with two products, right, one that's reacted with monoamine oxidase and COMT, and one that's only reacted with monoamine oxidase, and we take those two products and react those with two different enzymes, that means we're going to get 4 different products, and I hope that makes sense. So what we can get is the aldehyde on that is part of the alpha carbon. We can ultimately oxidize that using aldehyde dehydrogenase. And if we do that, notice that we get a carboxylate. And really keep in mind that this is at physiological pH, so it should be a carboxylate, not a carboxylic acid. But I think you get the idea, right? So if we take the aldehyde and oxidize it using an aldehyde dehydrogenase, and keep in mind that's an NAD-dependent oxidation, we should get a carboxylate, right? And that's characteristic of both vanillomandelic acid and 3,4-dihydroxymandelic acid, or we could say vanillyl mandolate or 3,4-dihydroxymandolate, right? Okay. And keep in mind that vanillyl mandolate is one of the products that we get if we've reacted the previous substrate with catechol-O-methyltransferase, right? So in other words, if we already had the methyl group on the hydroxyl group that's meta to the beta carbon, that's going to give us vanillyl mandolate, right? But if we were to take norepinephrine aldehyde, the one that's not been methylated, and react that with aldehyde dehydrogenase, we're going to get 3,4-dihydroxymandolate, okay, because we don't have the methylated hydroxyl group meta to the beta carbon, okay? And we're going to follow a similar suit when we react it with aldehyde reductase. And this is an NADH or NADPH dependent reduction, okay? And normetanephrine aldehyde, the one that's been methylated, is going to react with aldehyde reductase. And again, keep in mind that if we're reducing an aldehyde, we should get a hydroxyl group or an alcohol. And we do see that we get an alcohol in both of these products down here. Okay, we get an alcohol. Okay, but if we already methylated the neurotransmitter on the hydroxyl group that's meta to the beta carbon, we're going to get something called 3-methoxy-4-hydroxyphenyl glycol. Okay, and notice we have the alcohol attached to the alpha carbon, right? And the meta hydroxyl group has been methylated. Okay, but if we take norepinephrine aldehyde, the one that's not been methylated by acetylmethionine, and we react it with aldehyde reductase, notice what happens. We get the hydroxyl group still, right? We still have this, but we get a different product called 3,4-dihydroxyphenyl glycol. Okay, so I hope this makes sense. You can react the neurotransmitter, whether it's dopamine, epinephrine, or norepinephrine, with catechol-O-methyltransferase and or monoamine oxidase, right? But you have to react with monoamine oxidase to do total inactivation. And the total inactivation of the aldehyde is accomplished through aldehyde dehydrogenase or aldehyde reductase. And I hope that makes sense. Now, one thing that I want to make perfectly clear is this. Let's say that instead of reacting norepinephrine in this pathway, let's say we did dopamine. Well, keep in mind that in the case of dopamine, this group right here is just a hydrogen, right? That group, let me make that perfectly clear, this group is a hydrogen. It's not a hydroxyl group because norepinephrine is the product of dopamine and dopamine beta hydroxylase or dopamine beta monooxygenase, right? So dopamine just has that hydrogen there. It doesn't have the hydroxyl group. So in that case, what you would do if you wanted to predict all of these products for dopamine catabolism, right? What you would do is you would basically take this hydroxyl group, and in every product you would replace it with a hydrogen. And I hope that makes sense, because in dopamine you can react with all of the same enzymes, right? But that hydroxyl group will be absent. It'll just be a hydrogen. And I hope that makes sense, okay? But one thing I want to say about epinephrine is this. You might be tempted at first to say that epinephrine is going to give different products than norepinephrine, but that is absolutely incorrect. Okay, it's going to turn out that in the case of these products over here, in these 4 products that you get after the dehydrogenase and the reductase, okay, after you get those products, the epinephrine products will be exactly the same as the norepinephrine products. And we'll go into why that is in a few minutes. But what I want to do now is I want to actually look at the mechanism of catechol-O-methyltransferase, just so you can get intuition as to what it is actually doing. Now, catechol-O-methyltransferase, remember, can react with either dopamine, norepinephrine, epinephrine, or remember that it can react with the corresponding aldehyde once monoamine oxidase has dealt with it, right? So effectively what you could say is this group right here could in fact be an aldehyde. Okay, I've just simply drawn it as say that a catecholamine had reacted with catechol-O-methyltransferase. Okay, but that doesn't have to be the case. This group that's in orange could be an aldehyde, and I want to make that perfectly clear. Now what I want to do is really get a little knowledge about what catechol-O-methyltransferase is doing. Okay, now keep in mind that the only hydroxyl group that's getting methylated is this one. Okay, it's the one that is meta to the beta carbon, right? But it's ortho to this hydroxyl group that's right here. Okay, so that's the only one that's getting methylated. The hydroxyl group that's para to the beta carbon does not get methylated. So this enzyme, at least in the case of the benzene ring, it's absolutely specific for that hydroxyl group. It's not absolutely specific for the substrate, but at least in terms of catecholamines and degradation products, it's specific for that hydroxyl group. Okay, so what's effectively going to happen is there's going to be a base in the active site of the enzyme. Okay, there's going to be a base, and it's going to deprotonate. It's going to deprotonate that hydroxyl group. Okay, and what's going to happen is those electrons between the oxygen and the hydrogen are going to attack this methyl group that's on S-adenosylmethionine. And certainly, if you need help with the mechanism of methyltransferases, certainly go back and watch the video on that. But effectively, catechol-O-methyltransferase is going to catalyze the attack on that methyl group. And of course, you're going to have loss of the leaving group. And in the case of this enzyme, and all methyltransferases of this kind, the leaving group is just S-adenosyl homocysteine. So we can say it's adohomocysteines. And so you can say that this one is adomet, right? Adenosylmethionine or S-adenosylmethionine. And so what you should get at the end of this is you should get a methylated oxygen that is meta to the beta carbon, right? I keep saying that because it's really important when you're trying to predict products on an exam, okay? And also keep in mind that at the end we also should have our base in the active site that's protonated, right? Our base in the active site's protonated, and what will effectively happen is there's a theoretical step in which there will be a proton exchange with solution to regenerate the resting state of the enzyme. So I hope that gives you a little bit of intuition on what catechol-O-methyltransferase is actually doing. What I want to do now is get a little bit of intuition on monoamine oxidase. And for those of you who haven't seen this before, sometimes you'll see this as MAO, and oftentimes if you have an inhibitor of monoamine oxidase, they'll often say an MAO inhibitor, so it's kind of important to know. So MAO is monoamine oxidase, and by the way, there are multiple monoamine oxidases that have various specificities for different neurotransmitters, but we're just going to talk about it in general. Okay, now keep in mind this R group right here. If this is a hydroxyl group, then that's characteristic of both norepinephrine and epinephrine and their metabolites, right? But if that R is a hydrogen, then that's going to be dopamine. And if that R was a hydrogen, then this R prime would also have to be a hydrogen, right? Okay. And what this enzyme is going to do is it's going to use FAD in order to basically take this part right here, and it's going to oxidize it into a Schiff base, okay, or to be specific, a protonated Schiff base. Okay, and that's what the mechanism involves, and maybe one day we'll have an entire video on the mechanism, but suffice it to say for now, the mechanism is extraordinarily complex and convoluted, and as we know, Schiff-based mechanisms are not exactly the easiest thing in the world. Okay, they get really long and hard really fast. Okay, so we're not going to deal with that here, but suffice it to say that group will become a protonated Schiff base. And then what's going to happen is we're going to have water that's in the active site, and the water is going to do a Schiff base hydrolysis, okay? And that oxygen, let me do this in green, that oxygen that's part of the water, that's this oxygen that's part of the aldehyde, okay? So that's what we mean when we get a Schiff base hydrolysis. So when we do the Schiff base hydrolysis, okay, we're going to get an aldehyde as a result, and this would be the aldehyde that's this right here. Right, that would be these aldehydes right here, this one right here, and this one right here. So in the case of norepinephrine, that would be normetanephrine aldehyde and norepinephrine aldehyde, and that would depend on whether or not it's reacted with catechol-O-methyltransferase. Okay. But in this case, we're assuming that it hasn't reacted with catechol-O-methyltransferase. But we do the Schiff base hydrolysis, and we're going to get an aldehyde as a result, and I hope that makes sense. Okay, and that's all part of the mechanism of the FAD-dependent oxidation. So if FAD is oxidizing this right here into a Schiff base, right, then FAD in the process had to get reduced. Okay, so in the process of doing this, we're going to get an FADH2, and I hope that makes sense. Oxidations can't occur without reductions, right? So if we're oxidizing the catecholamine into a Schiff base, that's an oxidation, so FAD had to get reduced to FADH2. And again, the mechanism is very complex, very convoluted, and we'll look at that in another video sometime. Okay? But then the Schiff base gets hydrolyzed into an aldehyde, and then you get this molecule right here. Okay? Now this molecule changes. Okay? Now remember what I said earlier. Back to this picture. Okay? Dopamine products change, right? Because remember, in the case of dopamine, this was a hydrogen, right? And you can make this case in all the other molecules. That was a hydrogen in dopamine. So that product is going to be different than norepinephrine and epinephrine degradation products, right? But remember how I said that in general, these products right here, these products are the same in norepinephrine degradation and epinephrine degradation. Well, let's think about why that might be. Well, they both have this hydroxyl group on the beta position, right? But remember that the part of the molecule that gets hydrolyzed off is this, right? The amine. Now, in terms of the actual degradation products, which is this, right? Norepinephrine degradation and epinephrine degradation is exactly the same, right? And I think you can see that, right? But the amine that gets hydrolyzed off through the Schiff base hydrolysis is different, okay? Now, we'll come back to that in a few minutes, but let's actually talk about what happens to FADH2, okay? The reason this enzyme is called an oxidase has nothing to do with the first half of the mechanism. In fact, the first half of the mechanism is just an FAD-dependent oxidation. And in the process, remember, FAD gets reduced to FADH2. Well, the oxidase activity is going to use molecular oxygen in order to oxidize FADH2 back to FAD. And in the process of this oxidation, oxygen gets reduced to hydrogen peroxide. Okay, so this oxygen is going to get reduced ultimately to hydrogen peroxide. Okay, and that, of course, regenerates the resting state of the coenzyme FAD. And then we're ready for another cycle. Now, the hydrogen peroxide is going to get released by the enzyme. And as we know, catalase is going to degrade hydrogen peroxide into oxygen and water. Okay, and I hope that makes sense. Okay, we've dealt with that in other videos. Okay, but let's come back to the problem. Okay, in the case of dopamine, let's think about what this amine might be. Well, in the case of dopamine, what was our R prime? Well, in the case of dopamine, it was just another proton, right? So if we're dealing with dopamine, we'll abbreviate that DA because that's normally the abbreviation. Let's think about our amine product. Well, in the case of dopamine, our amine product is just going to be ammonia, right? Because that other R prime group was a proton, right? So in the case of dopamine, that's going to be ammonia as our product. And we know from our studies of amino acid catabolism that the ammonia will either get ligated to alpha-ketoglutarate to make glutamate, or from glutamate, ligated to glutamate to make glutamine, or if we're in times of amino acid catabolism, we're going to essentially put it into the urea cycle. So it really depends on the energy charge of the cell as to which direction we're moving. Okay, but the point is we're producing ammonia. Okay, now let's think about norepinephrine NE. That's normally how we abbreviate it. What's going to be our amine product? Well, when we hydrolyze off that amine in the shift-base hydrolysis, our R prime is also a proton. So if we do this process with norepinephrine, we're going to get ammonia as well as our amine product. But now let's think about epinephrine. What's going to be our product there? Well, what's our R prime group in epinephrine? Well, our R prime group is a methyl group, right? Remember that norepinephrine reacted with phenylethanolamine and methyltransferase, which was a SAM-dependent enzyme. And so that R prime is a methyl group, right? So that means our product is going to be this. Let me do it like this. Let's do this. So H3N and then a methyl group, right? Still has a positive charge at physiological pH. So we get methylamine. Now, we're not going to go in excruciating detail here about methylamine, but suffice it to say what's going to happen is there's going to be another reaction that is exactly the same mechanism as monoamine oxidase, okay? Basically what you're going to do is you're going to take the amine and oxidize it into an aldehyde. So let's think about what might happen if you oxidize that amine into an aldehyde. Well, your initial product is going to be a shift base, right? We're just doing the same mechanism that we did here, right? We're basically taking this group right here and doing the analogy down here. Well, if we oxidize this into a shift base, what do we get? Well, we're going to get this, right? We're going to get this, right? And I hope that makes sense. Oops, going to get this. So we get the shift base version of the methylamine. And of course, this is an FAD-dependent oxidation. It's the exact same mechanism, right? So what's our other product going to be? Well, our other product is going to be FADH2, right? Because in the process of doing the oxidation to the shift base, we should also get a reduced coenzyme, which is FADH2. Now, we already know what happens to FADH2 through the mechanism. You're going to use molecular oxygen, okay, and you're going to basically oxidize FADH2 back to FAD, and you're also going to get hydrogen peroxide. And we know that hydrogen peroxide will get released by the enzyme and degraded by catalase. But our shift base version of methylamine, which is 2 electrons short, what happens to that? Well, we know from our study of monoamine oxidase, this is just going to undergo a shift-base hydrolysis, right? It's going to do a shift-base hydrolysis, and let's think about what our products are going to be of that. Well, if you need help with predicting products of shift-base hydrolysis, certainly go watch the video on that. But if we were to do a shift-base hydrolysis of this, we're going to get 2 products. Number one, we're going to get ammonia. Okay, that's our first product. But we're also going to get formaldehyde, okay? And we're not going to go into detail here about how we degrade formaldehyde, but suffice it to say, there is a degradation pathway for formaldehyde. So one of the problems with degrading epinephrine is it's a lot longer of a catabolic pathway. And the reason is because we're producing this methylamine, which we don't want a lot of. Methylamine's not a good thing to have floating around. So we do another oxidase to oxidize it ultimately into formaldehyde and ammonia. And we know that the ammonia will either go into the urea cycle or it will get ligated to either alpha-ketoglutarate to make glutamate or to glutamate to make glutamine, depending on the energy charge of the cell. Okay? And by the way, the enzyme that does this, they're part of a large family of enzymes, and they're called amine They're called amine oxidases. And they're all going to follow the same mechanism as monoamine oxidase, because monoamine oxidase is part of the amine oxidases. It's just that the identity of this group over here is different. Okay? In the case of your catecholamines, it's all this catechol group in the beta carbon, right? But in the case of methylamine, right, it's a methyl group, right? So the identity of that group changes, okay? But the mechanism is exactly the same, okay? So we're going to oxidize to a Schiff base, do a Schiff base hydrolysis, and get ammonia, right? We're going to get ammonia and then some kind of carbon product. In this case, our carbon product is going to be formaldehyde, which will get catabolized further, okay? And it's actually going to involve things like glutathione and all that, okay? So I hope this gave you a little bit of intuition of what monoamine oxidase is doing. And keep in mind, it's just part of a large family of enzymes that are amine oxidases, okay? And again, monoamine oxidase, astronomically more complex than catechol-O-methyltransferase, right? But I hope this gave you intuition on what exactly the degradation pathway is doing, okay? And keep in mind, the most important thing about these pathways is really just to be able to predict the products. From there, you can sort of get good at naming them based on what it looks like, but the important thing is really to be able to predict the structure of the product. So keep in mind, you can react with catechol-O-methyltransferase and monoamine oxidase, or you can just react with monoamine oxidase. But to fully inactivate the product, okay, to fully inactivate the catecholamine, you have to have the aldehyde, which will either get dehydrogenated to form the carboxylate, which is in purple, or reduced by aldehyde reductase to form the hydroxyl group. Okay, and by the way, these products over here on the right side of this picture, they get excreted. Okay, they get excreted by the kidney, and also sometimes if you're at the epinephrine or norepinephrine or whatever gets released into the blood, okay, they can also get excreted. So it's not just like the degradation products are the only things that get excreted, okay? Sometimes the kidney can actually excrete the actual catecholamines themselves, not just the degradation products, okay? So I hope this video gave you a little bit of intuition on catechol-O-methyltransferase and monoamine oxidase. Again, the most important thing to be able to do is to predict the products. See you in the next video.